Hi everyone, and welcome to episode 21 of the Cancer Researcher podcast, brought to you by the European Association for Cancer Research. In this episode, we invited Professor Charlie Swanton, Deputy Clinical Director at the Francis Crick Institute and Chief Clinician at Cancer Research UK, to discuss his research interests and some challenges he faced in his early career ahead of his keynote lecture at the upcoming EACR Congress. I am your host, Dr. Alexandra Boitor, Scientific Officer at the EACR, and I invite you to dive into today's topic. Hi, Charlie, and thank you for accepting our invitation, or should I say invitations, first to give a keynote lecture at our Congress in June, and then to have a little chat with me for our podcast.
Hi, great pleasure. Thank you for inviting me.
It truly is an honor to have you here today. You are one of the most prolific and respected researchers in the space of oncology and medicine, and one of the living examples of building up on discoveries from fundamental research, but also of building up an impressive career. And on that note, you somewhat recently published a paper in Nature about your personal experience with your PhD, about making the best out of your circumstances. I think you've entitled it Turning a Failing PhD Around. So could you please share with us that story of what you went through with your PhD?
Right, okay. So I did my PhD in the mid-90s at what was called the Imperial Cancer Research Fund in Lincoln's Inn Fields. This was an extraordinary place. It was a sort of a melting pot of scientific discovery and, had been the place of birth of many very important discoveries in fields like cell cycle, discovery of oncogenes, understanding of cellular apoptosis and cellular signaling. And it was an incredibly vibrant place. We had a phenomenal seminar program. Doors were wide open. You could go in and out of people's offices, very senior scientists, and ask some questions about your research. And people were only too happy to help. It was an extraordinary environment to work in. So I started as a PhD student there in 1994, and I wanted to study the cell cycle. One of the original questions I started to ask was, why were there three different cyclin Ds? There's a cyclin D1, D2, and D3, and two different kinases, CDK4 and CDK6. And it was a reasonable assumption back then that the reason there were three different cyclins binding the same CDK was that the cyclins conferred substrate specificity to the kinase, which they do. But the idea was that perhaps different cyclins could bind different substrates. And so I set about making these so-called chimeras between cyclin D1 and D3 and splitting their domains up and fusing them. So I had sort of cyclin D1, D3 chimeras and cyclin D3, D1 chimeras, etc. And I had to show they bound the kinase and then that they perhaps influenced the phosphorylation of substrates differently. But unfortunately, after about 18 months, I realized they didn't. And I even struggled to get positive controls working. And I was banging my head against a brick wall, essentially, not really getting very far. But what I did know was this scientific environment that I worked in, where I could walk into anybody's office and ask them questions about the science, about the experimentation and the systems I was working in, and seek their advice. And, these are world-class scientists who had discovered many components of the cell cycle. And it was just such a privilege to be able to walk into their office and talk to them about the challenges I was facing, and they would give me time. I just thought this is just an amazing way of running one's working life. This didn't feel like work. I looked forward to going in on a Monday morning, and I was essentially paid to pursue curiosity-driven research. What an enormous privilege. And it seemed like, frankly, a bit of a minor problem, the fact that my PhD wasn't really working. I knew I was enjoying it. Anyway, the crunch time came and I had to sort of face facts that I hadn't really got very far, and I hadn't even got chapter 1 by month 18 of my PhD. In fact, I had really got a single experiment to work at all. The time came for me to hand in my midterm report and I didn't really have any data, so I was sort of delaying. Anyway, the graduate tutor at the time came into my lab and said, "Where's my midterm report?" And I said, "Well, I haven't written it yet because I haven't got any data and I'm waiting for an experiment to work." And he understandably got quite cross with me and insisted that my midterm report had to be on his desk on Monday morning, otherwise there would be consequences. So I went back to write it up and handed in what was frankly a pretty appalling midterm report. And that resulted in a fairly urgent thesis committee review where they said to me, "Well, Charlie, you've got two options. One, you go back to medical school and complete your medical training and give up on a PhD. Or you can change PhD projects." I said, "Well, I'm not going back to medical school because I love this too much. I'll change PhD projects." And they said, "Well, what do you want to do?" I said, well, what I would really like to do is understand why the CDK inhibitors, or how the CDK inhibitors, P21 and P27, bind the kinase and the cyclin. And they said, how are you going to do that? I said, well, I'm not really sure. Came up with a few ideas, tested them, and they didn't work. And in the end, I did it the laborious way by mutating every cell surface exposed amino acid on cyclin D into an alanine until I found one that didn't bind P21 and P27, and cyclin still bound the kinase. And from there, everything fitted into place, because round about the same time, Chang and Moore had cloned the Kaposi sarcoma herpes virus that encoded a cyclin that was homologous to cyclin D. And it turned out that that one domain that I found that could interact with P21, P27 had been mutated on the viral cyclin. And I did an experiment that showed exactly that the viral cyclin had evolved to mutate those cell surface residues so that it could only bind the kinase, but not the cell cycle inhibitor. And so it was a dominantly active cyclin that could enable the viral genome to be replicated efficiently without the breaks of the human cell cycle system from inhibiting the cyclin CDK complex. And for me, that was just a light bulb moment. It was just the most incredible moment, realizing that was the case. You've seen something that no one on planet Earth had ever seen before. And I thought, right then, I want to do this for the rest of my life. This is the job for me. Of course, those moments don't happen very often in science, but when they do, oh my God, they're so exciting.
That is indeed inspiring, your perseverance, but also to think that your research journey started from the sheer curiosity of understanding how P21 interacts with other proteins. And now you are one of the chief investigators on lung cancer in the Tracer X project and are involved in clinical trials as well. Fascinating and truly inspiring. You mentioned having a choice between going back to the medical school or switching gears with your PhD project. And we well know you are a physician scientist, so naturally I must ask what drew you specifically towards cancer research?
Yeah, so when I was at medical school, I got a phone call at the end of my first year from my father, who said that he'd lost sensation between his legs, something called saddle anesthesia, which is sort of very indicative of a lower spinal problem, often spinal tumors. And that's indeed what he ended up having. It turned out the spinal tumor was a high-grade B-cell lymphoma. And he had chemotherapy and radiotherapy over about 8 months, very intensive therapy. And, 30 years later, he's alive, fit and well. And I thought back then, when I got that phone call, we didn't know what this tumor was. They thought it was a much more aggressive tumor than the one it turned out to be, or at least they thought it was going to be an incurable tumor. But the biopsy showed otherwise, and it showed it was a lymphoma that is curable potentially with chemotherapy, and now more recently, rituximab, a monoclonal antibody against CD20. So I thought back then, this was a terrible year. He was very sick, both due to the tumor, but also due to the therapy. The doctors looking after him and the chemotherapy regimens they were using saved his life. And I thought to myself, well, how did this all come about? And it's obvious how it all comes about. It comes about through meticulous discovery research that leads to new cancer drugs, that lead to randomized phase 3 trials that either show improvement or not. And each step of improvement incrementally improves survival modestly, but taken together over decades of research leads to cures. And I thought to myself, I wanted to be part of that one day. Cancer was clearly the greatest unmet need when I was at medical school. We'd made great inroads in cardiology. Oncology seemed to me back then to be one of the greatest challenges, and still is. I think that, along with neurodegenerative disease, are the two sort of biggest challenges that face discovery and clinical researchers now. I'm very fortunate to have been allowed to work in this field, both as a medic and as a scientist funded by Cancer Research UK over the best part of 22, 23 years. It's been an extraordinary journey, seeing the way in which just in those 2 decades, cancer survival times have improved in several diseases, how our therapeutic regimens have advanced, how immunotherapy is now the mainstay of the management of many solid tumor diseases. And really also seeing the way in which technologies have advanced that have enabled us to open a window on a world of molecular biology and cancer that's revealed sort of the inner intricacies of cell division, cell communication, and most importantly, how the immune system interacts with an evolving tumor. I think it's simply staggering what we've learned in a very short time from advances in the community.
You've half answered my next question because I wanted to ask you, what would you say are the major changes you noticed in the cancer research field since you started working in it? And I'm interested in finding out your opinion both on research topic evolution, which you've briefly touched on, but also on how the life of a researcher has changed over the years.
Yeah, great question. So I think one of the biggest changes in the last 2 decades is this explosion in data, which is a good thing in many ways, but also presents new challenges. And I think that should come potentially with a health warning, which I'll explain in a minute. But when I first started as a discovery scientist in the mid-90s, bioinformatics didn't really exist as a specialty. And certainly there wasn't the sort of level of data that there is now. And to some extent, that's been generated by new technologies such as next generation sequencing, which has created this explosion in data in terms of DNA and RNA sequencing, and more recently, ATAC-seq and other technologies. And actually, we've got more data than we have scientists to analyze it. And along with that, of course, is deep learning, artificial intelligence, machine learning tools that are further illuminating biology in ways in which we could never have imagined 2 decades ago. So if you look around an institute like the Francis Crick Institute where I work and think to yourself, well, how does it differ now compared to 25 years ago? Without a doubt, 25 years ago, there would have been many, many more people at the bench than there are today. Many postdocs are sitting in front of a computer processing data now because there's so much more data than there was 2.5 decades ago. And also because the technologies have advanced to such an extent, there's a need to have experts that run these technology cores. And so what I'm also seeing is, for instance, at my institute, we have over 15 technology platforms. So these are laboratories that run, for instance, light microscopy, cryo-EM, flow cytometry, advanced sequencing, etc. And they're highly specialized technologies that need to be run by people doing this full time. And so what I'm seeing is that postdocs have a sort of menu of experiments that they're almost contracted out to these laboratories. And postdocs and PhD students act in many ways like conductors, conducting broad experimentation to address their question, utilizing these different scientific technology platforms. And that certainly wouldn't have happened at that scale 25 years ago. So what about research topics? Well, the obvious research topic that I think I've mentioned already to some extent is this field of cancer immunology. In the mid-90s, funders were abandoning cancer immunology as an area to develop because they'd largely given up on this idea that the immune system could be used as a tool to fight cancer. Well, of course, we know, thanks to Honjo and Jim Allison and many others, that that's clearly not true, that there were very fruitful approaches. We just needed to look harder. And of course, the finding of CTLA-4 and PD-1, these sort of breaks on the immune system, have opened up a whole new field of cancer immunology that have led to cancer vaccines, checkpoint inhibitor therapies, CAR T-cell therapies, the modification of T cells to target tumors, as well as biological therapies like bispecific antibodies, antibody drug conjugates, and what have you, that essentially use the immune system or components of the immune system to target tumors more effectively. And that's been incredibly exciting. In my field, I think the area that changed the most for us is sort of understanding now that cancers are evolutionary phenomena that are constantly subject to random variation due to genome instability, chromosomal instability, and point mutagenesis. And that provides a substrate upon which natural selection acts, and selection in a tumor can manifest in many different ways: drug pressure, hypoxia, radiation therapy, predation from cells around the tumor as the tumor's growing, being starved of resources and what have you. And this evolutionary force plays out across the disease landscape from diagnosis through to either cure or death. And understanding cancer as a sort of evolutionary phenomenon, I think, is helping us design trials more effectively. It's helping us design drugs more effectively, and biomarkers and mechanisms that lead us to detect tumors earlier as well. And I think that's been the sort of biggest change in my view, and the way in which we run our lab is thinking about cancer as an evolutionary and adaptive conglomerate of different cell types.
Switching gears in our conversation, I would like to ask you about your own research. I mean, you have an undeniable talent for bringing different research topics and fields together. Your lab works on cancer evolution and genome instability, covering issues such as tumor heterogeneity, clonal evolution, resistance to therapy, bringing together functional cell biology with cancer bioinformatics and clinical trials, elements of cancer prevention, as seen in your recent papers looking at the impact of air pollution on lung cancers, diagnostic and treatment. You really do seem to cover the entire spectrum of cancer research through your work. So what I'm really curious about is, what is your favorite research question or project that you had the opportunity to work on?
Right, gosh, that's quite a question. I think probably the field that I'm most excited about right now is this area of molecular cancer prevention. So, what do I mean by that? Well, thinking about a future a little bit like cardiovascular disease, where we can offer a patient a long term treatment like a statin to prevent patients getting cancer. So, why is now the time for that type of approach? And I think it's because really of seminal work from people like Alan Belmain and before him, Isaac Berenblum, who posed this question: how do cancers start? And the model of cancer initiation that I was always bonded to was this very simple classical model where carcinogens cause cancer by mutating DNA. Mutant DNA results in driver mutations, which expand clones into subclones, which result in secondary and third and fourth oncogenic mutations or tumor suppressor gene mutations that lead to a frank cancer developing. Well, it turns out that Alan Belmain and colleagues have shown that most carcinogens don't mutate DNA. So how are they causing cancer in rodent models in that case is the big question. And so Alan and colleagues have been really pioneering forces behind this idea that Isaac Berenblum proposed in 1947, that cancer is a sort of initiation as a two-step event. And actually, it wasn't just an idea. He underpinned this with very elegant mouse experiment work, where he showed that cancers require two steps: an initiating step and a promoter step. We now know the initiator step as a result of pre-existing mutations in normal tissue that occur just by consequences of aging, from wonderful work from people like Peter Campbell, Inigo Mascarenhas, and others at the Sanger, showing that we're essentially a patchwork of mutant clones. James de Gregory, for example, estimates that in a human body of a 60-year-old, there are 100 billion cells with a mutated oncogene. That is a phenomenal statistic. And that tells us that cancer at the cellular level is incredibly rare, given that we're all a patchwork of mutant oncogenic clones. And so these mutations sit there for the lifetime of the patient and often don't cause any problems at all, usually don't cause any problems. So something else is required, and that's what Isaac Berenblum referred to as a promoter, that we know in some cases can be an inflammatory process. We've shown in a context of air pollution, for example, part of that inflammatory process we think is mediated by interleukin-1 beta, with very good evidence from the CANTOS trial that antibodies against interleukin-1 beta can actually prevent lung cancer from initiating. So we're super interested now in trying to understand how these carcinogens drive tissue inflammation that contributes to the promotion of these pre-initiated cells that occur in normal tissue with oncogenic mutations. And I think that represents a formidable challenge, but also a very exciting one that could lead to new therapies that can actually prevent these mutant clones from evolving into frank malignant disease.
That sounds so interesting. I can't wait to find out more. Are you planning on sharing some of the data you have on this project with us in June as part of your keynote lecture?
Yes, I absolutely am. This is exactly what I'm going to do. Yes.
There's only a few months left now then. So if you're interested in the latest developments on Professor Charlie Swanton's research, join us for the EACR Congress in June in Lisbon. Early rate registration deadline is on 28 April 2025. Check the program at 2025.eacr.org. So as I was saying, your keynote lecture is most certainly one of my highlights for the Congress to come, especially after the little teaser that you've just given us. But to reverse the question, could you share with us one thing that you are looking forward to at the EACR Congress in June?
Well, I think what I look forward to most actually is interacting with colleagues, particularly the junior colleagues who will be at this meeting. I often find the best questions, the most challenging questions, come from those who are newest to the field. And I think the reason for that is because they approach the field in a very unbiased way with no preconceived ideas. And I think that's the beauty of science in so many ways. It's that it's non-hierarchical. A PhD student or a master's student has as much to contribute to the scientific endeavor as a PI who's been in the field 30 years. And it's always those questions at the end of the lecture that stump me, that make me think we need to address this when I get back to the lab, when I leave Lisbon and get back to London. And I think it fuels discoveries. It fuels the curiosity that essentially keeps us in this job. We are always challenged by our data, and there are always flaws in it, in all our data. And I think being able to talk to people frankly about our data openly at conferences like EACR. It's a real opportunity to get real-time feedback from junior and senior colleagues alike and address those experimental flaws when I get back to London over the next year or two. It's a constantly evolving field and one that we constantly need to be on our toes about to address and provide the highest quality data possible.
And that actually brings me to my last question. So you said you're looking forward to networking with your colleagues in the field. But whilst this might come natural or relatively natural to more senior scientists, it is often quite challenging for early career researchers. So I was wondering if you've got some piece of advice to encourage them or to help them network and ask questions at conferences in general and at our Congress in particular.
Yeah, I think that's a great question. I think, first of all, don't be shy. I think there's always a tendency to not ask a question if you're in the audience because you're worried you might embarrass yourself. I would say there's rarely a stupid question in science. And often the most stupid questions you think are the most stupid are the questions that scientists either haven't thought of or they've thought of but can't answer and stumble them. So I would encourage everybody to speak up, to ask questions if they don't understand the logic, ask for clarification. And most importantly, at the end of the talks, go up to the speakers and talk to them and introduce yourself and ask them yourself what's perplexing you or offer up ideas for the next experiment. I'm always super impressed when junior faculty or PhD postdocs, master students come up to me at the end of a conference or at a poster or at the bar afterwards and ask me a question. Very often I won't have a good answer, or very often I won't even have thought of the question. And that's as instructive for me as anything. It's extremely helpful getting feedback from people perhaps new to the field. I'm always very impressed when the juniors have the boldness and bravery to introduce themselves and ask questions and put themselves out there. And that's what science is all about at the end of the day. So just don't be shy and enjoy it and accept the fact that in all likelihood, the scientist you're talking to won't know the answer to your question.
Thank you. I'm sure your advice puts some early career researchers' minds at ease a bit and hopefully encourage them to be more interactive when they go to conferences. Thank you very much for your time, Charlie. We really appreciate it. We know you're really busy. And thanks again, and looking forward to meeting you in person in June.
Lovely to talk to you. Thanks so much.
Thank you.
Bye.
We really hope that you enjoyed listening to this conversation. I would like to thank again our guest, Professor Charlie Swanton, for being here today with me, and many thanks to everyone who listened. You can find out more about the EACR Congress, including information about the Congress program and associated events such as Cancer Researchers to Watch, an early career showcase, and the EACR Women in Leadership workshop by accessing the EACR Congress website at 2025.eacr.org. The early rate registration deadline to the EACR Congress is 28th of April 2025 and provides EACR student and early career members eligibility to register for the Early Career Showcase. Applications for the EACR Women in Leadership workshop are open until the 22nd of April 2025. Please subscribe to our podcast so you won't miss an episode. You can find The Cancer Researcher on your podcast platform of choice. Subscribe and tell your friends about it. You can also find us on Instagram and Blue Sky at Hello EACR, and you can search for us on Facebook and LinkedIn. As always, we welcome feedback and suggestions through our feedback form, which you can find on our website at www.eacr.org/podcast. I hope to meet some of you at the Congress in June